if you open the incubator today, you do so at your own risk.
Yeah.
It's really pretty nasty. I will take pity on you tomorrow and come in early and go take the gas pack and the candle jars outside and open them up, because in those particular environments you get some interesting smelly organisms. So tomorrow in lab we're just going to be looking at all of the results, recording all the results. You'll be taking pictures of your glycolate tubes, of your gas pack versus aerobic control, of your candle jar versus aerobic control, and then you will have the some of the known organisms for kind of reference. And then again, you know, if you've got things you need to be doing on your pure culture exercise, you can work on that as well. Next week, we get to do some throat swabs with one another and see if you're carriers for Staph aureus. So if anybody has a gag reflex, this will be a fun lab for you. We'll do some skin swabs too. Any questions? Okay, I asked you last time to kind of look through the terminology. I'm not going to go through all that. It's written out for you, but I will assume as we're talking, you understand the difference between things like a disinfectant and antiseptic, a chemotherapeutic agent, a sanitizing agent, et cetera, et cetera. So if I'm using those terms and you haven't looked at this list yet, you should. Make sure you understand the vocabulary. So as I said, this is a pretty good organizing slide for keeping track of some of the things that we're going to be talking about. We're really going to kind of work our way through, starting with physical agents of heat and radiation. We'll then pop over to the right hand of the side and talk about some mechanical agents for controlled microbial growth, primarily filtration. And then we'll get into some of the chemical agents that can be used for disinfection, sterilization, sanitization. In this particular set of information, we're not going to be talking about chemotherapeutic agents yet. Chemotherapeutic agents are the ones that are designed for internal use. And so we'll be talking more about things like bleach and chlorine and things like that here. And then in the next material, in the next chapter, there's a set of PowerPoint slides and lecture notes for chemotherapeutic agents. So that's when we start talking about some of the antibiotics and various other agents that you can take inside of the body to try to stop microbial growth. We saw that there were a number of factors that could influence microbial growth. And by the same token, there are a number of factors you've got to consider when you're trying to kill microorganisms. Okay? What are some of those? Well, one of the considerations is what type of bacterium are you going to try to be destroying? What's probably one of the biggest considerations in terms of species difference? Some organisms have it, some don't. In terms of the structure that really determines whether an organism is easily killed or not. Gram-positive, gram-negative could be one of the differences, and that's a reflection of cell wall differences. Then when you're talking about cell walls, if you're acid-fast, that glycolic acid that's present in the cell wall could be a consideration. What were you going to say here?
Capsule?
Capsule, possibly. That's probably going to be more of a consideration when we start talking about some of the chemotherapeutic agents. Okay? Any other cellular structures that might influence the susceptibility of an organism to various control mechanisms? Endospores, absolutely. Yeah, that's really a big one. And whether those bacteria are spore-forming bacteria or not. But even in the non-spore-forming bacteria, there are fundamental differences in the genetics of those organisms which do influence their susceptibility. So one of the things we're going to see is different control methods are oftentimes tailored to a particular pathogen that you're concerned about. And I'll give you a couple of examples about that as we go along. When we are autoclaving, for example, one of the things that you've really got to be concerned about in autoclaving is you don't overstuff the autoclave with things. How much stuff you put in an autoclave is referred to as its load. Okay? And so the greater the load of the autoclave, typically the more time you've got to autoclave to achieve sterility. So you can see that on this particular slide right here. The time of sterilization really varies depending upon whether you have a high load in the autoclave or a low load in the autoclave. Time obviously is a factor that influences whether you've achieved sterilization or disinfection, depending upon what your goal is. This shows you a plot that relates the log of number of viable cells to the time of sterilization in an autoclave. And typically, when you get into this 12, 14 minutes range, that's pretty sufficient amount of time exposure for the autoclave to do its thing. And you can see over here, if you look at this particular graph, that's even at a relatively high load. But standard autoclaving time is not that low. Typically, we autoclave for somewhere between 15 and 20 minutes. And basically, we're just protecting ourselves, okay? I mean, we should achieve pretty much full sterility by the time you get down to about 14 minutes. But giving that extra couple minutes is just good margin of error to make sure that you've had sufficient time. And then that way, if there are some particularly resistant organisms you're trying to get rid of, you'll cover those as well. This particular slide here shows you the relative resistance of cells that are spore formers versus cells that are just vegetative cells that can't form spores. The type of agent that you're trying to treat the organism with, particularly if it's a chemical agent, can be a big consideration. Some agents are considered to be microbiostatic, where others are microbiocidal. What does static mean? Unmoving? Yeah. Inhibited?
Does it stop any future growth?
Yeah, it basically just inhibits growth, but it doesn't kill the microorganism. What does microbiocidal mean? What does cidal mean? Cidal means to kill. Okay, so static is inhibit. If you're put in stasis, for example, you're kind of in suspended animation, you're not dead. Versus cidal, you're dead. So if you look here at the effect of the application of a microbiostatic agent, basically the growth just stops and the organisms are just kind of in this stasis, this equilibrium. But if you apply a microbiocidal agent, you actually get killing and a decrease in the population. So the type of agent you're treating it with is important. When you're talking about the type of agent you're treating with, you also have to know what concentration you're applying. Because sometimes the same chemical agent in a low concentration can be microbiostatic, inhibitory, but at a higher concentration it can be microbiocidal. So knowing your concentration is really pretty important. For chemical agents, one of the things that can really complicate matters is the presence of organic matter. Organic matter can quite commonly serve as a chelating agent. You know what a chelating agent is? Chelation is? Chelating agents bind to other compounds and make them unavailable or inactive. And so basically dirt can inactivate antiseptics that you put on injured skin, for example, to try to kill the contaminating microorganisms. And the presence of the dirt chelates or binds the agent and makes it unavailable to affect microbial growth. So when you're told to wash out cuts well, yes, you want to try to wash out any potential contaminating bacteria that might have been exposed to your internal tissues. But the presence of that dirt too can also compromise then any chemical treatment like an antiseptic you might put on afterwards. Chemical agents sometimes are temperature dependent. Sometimes elevating the temperature can enhance the effectiveness of the chemical agent. So at low temperature, it doesn't work as well. At a higher temperature, it works better. pH can also be a factor that determines the effectiveness of chemical killing agents. Bleach, for example, is more effective at killing microorganisms and fungus at an acidic pH than it is a basic pH. So there's a lot of things you've got to kind of consider in order to come up with the best situation for a particular application for using a particular method to kill a particular type of microorganism. So where we're going to start is, we're going to start with heat over here. And we're going to first contrast the difference between dry heat and moist heat and give you some examples of those particular types of heat methods that we commonly use for killing microorganisms. Now, one thing it is important to know is that in general, most of these killing agents, particularly heat, rely on denaturation for achieving their desired goal. And denaturation of proteins in particular, to some lesser extent, denaturation of nucleic acids, is kind of what you're trying to achieve when you heat microorganisms to try to destroy them. Some compounds, like heavy metals, can also inactivate proteins and enzymes, but they don't do so by really denaturing the protein. Remember, the concept of denaturation involves a change in the tertiary structure of the protein. And remember that structure determines function, so if you unfold a protein, it doesn't do what it's supposed to do. An enzyme loses its ability to catalyze reactions.
So is there another like metal like inactivating?
Yep, absolutely. Like over here, that metal could then bind to the protein and inactivate an active site and block the substrate from binding to the active site. A lot of metallic compounds, gold and silver, work this way. I mentioned earlier that they have some antibacterial properties. Iodine is another compound that typically can bind to proteins and inactivate them by blocking active sites. But the loss of protein function is really pretty critical in most of these particular methods. When it comes right down to it, denaturing or inactivating proteins is what most of these methods are all about. Okay, there's a couple of concepts that you need to be familiar with. Because they give us some comparative points of reference. And the two that I'm going to be most interested in are thermal death time and thermal death point. Thermal death time is the shortest amount of time at a particular temperature to kill all of the organisms. So it's the least amount of time at a selected temperature. Now you might ask yourself, well, what governs selecting the particular temperature? Well, many times this is really relevant in the food industry. Because you can't just heat food randomly and make it as hot as possible to kill microorganisms, because what's that going to do to the food? It's going to degrade the food. It's going to change its flavor. You know, milk is going to curdle, for example. Absolutely, absolutely, yeah. I mean, that's what's happening when milk is curdling, is it's denaturing that casein protein in milk. Yep. So you really have to be careful when you're trying to come up with particular conditions to treat food, for example, that the temperature is not such that it causes an adverse effect on the flavor or the quality of the food. Thermal death point is the lowest temperature to kill in a 10-minute time period. So if you look at these particular organisms on this list right here, there's a variety of things including non-spore-forming bacteria, fungal spores, yeast, virus, protozoans, worm eggs, etc. And these are the assigned temperatures, and then here is the time that it takes to kill those particular organisms at those particular temperatures. And you see it's kind of all over the place, indicating again the variation that exists in susceptibility of organisms to different particular treatments. This is an informative slide because this really points out the importance of endospores and how endospores can really complicate your treatments to try to destroy microbial growth. Okay? So if we compare endospores and vegetative cells, then we can see their relative resistance over here of one to the other. So for example, moist heat methods, which we'll talk about in a minute. An endospore, it takes 120 degrees Celsius of moist heat to achieve killing power. Vegetative cells are killed at 80 degrees Celsius. So that endospore confers real heat resistance. It's about 1.5 times the relative resistance. Look at the resistance to radiation that's conferred by endospores, about 4 times. To sterilizing gases, about 1.5 times again. To various sporicidal liquids. A sporicidal liquid is a chemical that inactivates spores. 18-fold differences. So it's really important to know when you're treating things, are they spore-forming or are they non-spore-forming? Okay, so looking at heat, there's really 2 major categories of heat. There's moist heat and there's dry heat. And dry heat basically is either baking, such as in an oven, or incineration. Now, what are you doing here? Well, with incineration, basically you're just burning up the cells and destroying them that way. That's what you're doing every time you flame the loop of your inoculating loop or needle in your Bunsen burner. Bunsen burner gets to be about 1800 degrees Celsius, right in that sweet spot of the blue flame. And that just completely oxidizes everything, and oftentimes you'll see a little bit of ash on your loop if you've got too much stuff on there. Okay. At lower temperatures, it's a little bit more confusing about how heat actually kills. One of the things that heating does, obviously, is dehydrate cells. But that in and of itself is probably not the only thing that's going on. Because remember, one of the reasons endospores are more resistant is that they're very dehydrated compared to the vegetative cell. In the absence of water, proteins don't denature very well in heat. It's really much more effective to denature a protein if they're in an aqueous environment. So some other stuff is going on there when you start baking things for prolonged periods of time that is actually killing the cells. And we're not really sure what all of those things are. But one of the things that I do want you to take home from this particular slide is look at the efficiency of moist heat, like steam, versus dry heat, like an oven, in terms of killing microorganisms. Moist heat is much more effective at transferring thermal energy from one body to another than dry heat is. Okay? If you walk outside in Arizona in an air temperature of 104, you're going to be uncomfortable. but it's going to take a long time for your skin to heat up in that 104 degree air temperature. If you get into my hot tub set at 104, you immediately feel that heat uncomfortable. Okay, that's a pretty uncomfortable temperature because the water is much more effective at transferring heat energy than air is. So if you look at the relative comparison here, moist heat at 121 degrees Celsius takes about 15 minutes to sterilize. And when you say sterilization, does that involve killing endospores as well? Yes. That's part of the criteria for something being sterilized is endospores are destroyed. Look how long it takes at dry heat at the same temperature. 600 minutes. Okay? So that relative efficiency of moist heat versus dry heat is really quite important. As far as moist heat is concerned, there are several forms of moist heat treatment, and one of them is just boiling. Typically, if the cell is just a vegetative cell, boiling something for about 30 minutes is considered to be sufficient to destroy the vegetative cells. That won't kill the endospores, because boiling only achieves the temperature of 100 degrees Celsius. So when you get a boil water alert, for example, if there's been some break in a water main, some potential sewage contamination, that will disinfect the water because it will kill the vegetative cells, but it will not sterilize the water because it's not going to kill the endospores. Fortunately, most of the pathogens that they're worried about in a situation like that are fecal bacteria, and those are non-endospore forming bacteria. So you're pretty safe if you boil water, but it's not sterile. Steam, which is not under pressure, is another form of moist heat. Sometimes the steam is referred to as free-flowing steam. It's not contained in a vessel like an autoclave. You just let steam go over the object that you're trying to treat. If you expose something to free-flowing steam for 3 successive days, that process is called tyndallization. And that will get you close to sterility. Why 3 days? What's the strategy going on? Why not just 1 day? Free-flowing steam is like boiling. You'll get a temperature of about 100 degrees Celsius. So if you expose something to tyndallization, there's a device called an Arnold sterilizer that does this. Why can't you just do it 1 day? What are you not going to wipe out? Endospores, okay? If those endospores survive, then they're going to germinate and get vegetative cells. What's going to happen to the vegetative cells on the second day? Those vegetative cells that germinated from the endospores are not now going to be killed by the steam. But there might still be a few endospores around. And when they germinate, on the third day, they should be destroyed. Okay? So the idea is that on 3 successive days, any surviving endospores, when you cool it down, will germinate, and in the next day of exposure, we'll get those vegetative cells. But you can't get every last one, and so that's why it achieves close to sterility, but isn't technically probably still sterile. Why would you do this? Well, there are some materials that are quite heat sensitive that you just can't autoclave. Vaccines that are grown from virus and eggs are treated by tyndallization because they're very heat sensitive. There are some drugs, there are some foods that are treated this particular way because they just can't tolerate the temperature of autoclaving steam under pressure. Now, autoclaving is going to be, we have one of these, but we don't use it very often. This is actually a tabletop incinerator for your loop. The problem with these is you really can't sterilize the neck of tubes with these, and that's why we still use open flame. But our method of choice, obviously, is the autoclave for sterilization. We use the autoclave to sterilize virtually all of the media that we're going to use in the class, so tryptic soy agar, tryptic soy broth, the fermentation broths we're going to use later on. There are a couple of things that are heat sensitive that we have to treat by filtration, which we'll talk about later on. But basically what's going on in an autoclave is you are enclosing water within a sealed vessel, turn it up to heat, and as the water is exposed to higher and higher levels of pressure, that boiling point of water is elevated so that at 15 pounds per square inch, for example, the water doesn't boil at 100 degrees Celsius, it reaches a temperature of 121 degrees Celsius. An autoclave is really the same thing as a pressure cooker. I don't know if anybody's ever even seen pressure cookers anymore. It used to be very common for canning vegetables, for example. You might have seen your grandmother or somebody using a pressure cooker. One of the things that we commonly have to do, and I do this about just once a year at the beginning of the micro course, is you have to check your autoclave to make sure it is achieving sterile conditions. And just because the pressure gauge says 15 psi and the temperature gauge says 121 degrees Celsius, what if those are off? Okay? What if they've gotten out of calibration? And so there is a biological method you can use to test the ability of your autoclave to achieve desired results. And that involves these little vials you can buy. The little vials contain a spore strip with endospores from a very highly resistant form of bacteria. These are bacteria whose normal temperature optimum is like 50 degrees centigrade. So their endospores are among the most resistant we know. Within this vial is a potential growth media for those bacteria. And it also contains a pH indicator. In this case, it's phenol red. So basically, you throw these vials in the autoclave, and they should destroy the endospores. You then crush the vial, and that exposes the spores to that growth media. And if the spores have been killed, then there will be no change in color in the growth media because they're not fermenting any of the nutrients in the media. But if the endospores did survive, then they will ferment the sugars in the media, produce acid, and that acid will lower the pH and change the pH indicator to a yellow. So in this particular case, this would be a failed test for your autoclave. And you'd have to call someone in and say, okay, what's going on? So doing that kind of quality control is very important to make sure your instruments are working properly all the time. Another type of moist heat treatment that has a lot of applications is pasteurization. And this was obviously named from Louis Pasteur. Pasteurization does not give you sterility. Pasteurization is devised to set up a particular set of conditions of time and temperature to exposure to heat. And the goal is not to kill everything, but to kill certain targeted pathogens. One of the things that we commonly pasteurize is milk. Are there bacteria in milk? Yes, there are. If you leave milk out too long, or even in the refrigerator too long, what happens to it? It spoils, it sours, it curdles. Why is it curdling? Well, there's a lot of lactobacillus bacteria, typically Lactobacillus lactis, naturally occurring in milk. And those particular bacteria will ferment the sugar in milk. What's the sugar in milk? Lactose, okay, milk sugar. And produce acid, lactic acid, and that causes the milk to sour. That denatures the proteins, it precipitates the proteins. Okay, well that's annoying, but Lactobacillus lactis isn't going to kill you. Okay? Now, if you can knock down the numbers of those bacteria in the milk, that's going to increase its potential storage life. But what we're kind of concerned about, actually a lot concerned about in milk, is a bacterium that's a rickettsia called Coxiella burtneti. And the name of that particular organism isn't particularly important, but it can cause a milk-borne disease that can be deadly called Q fever. So pasteurization was designed specifically in mind to come up with a set of conditions that would effectively kill Coxiella in milk. Now, pasteurization techniques have kind of evolved. One of the first pasteurization treatments for beverages like milk was called batch pasteurization. And this particular pasteurization technique involved treating milk at 63 degrees centigrade, a very particular temperature, for 30 minutes. Why not 10 minutes at 100 degrees Celsius? That's going to destroy the milk. Okay? So this was found to be kind of the best compromise between killing the microorganisms and food quality. Later, flash pasteurization was developed, a higher temperature, but for a much less duration. Then ultra-high temperature pasteurization was developed, a much higher temperature, but again a lower time. In the last couple of decades, ultra-high temperature pasteurization has achieved sterilization if you expose for 140 degrees Celsius for 1 to 3 seconds. Anybody ever seen on the store shelves milk sitting on the aisle as opposed to the refrigerator case? Oftentimes it's in boxes. This is very common in Europe. We were in Europe 10 years ago, and the grocery store shelves were just filled with this ultra-high temperature sterilized milk. And you can really taste the difference. Okay? So that exposure to that higher temperature does have an effect on the quality. But if you're used to drinking that, that tastes normal to you as opposed to our refrigerated ultra-high temperature pasteurized milk. So there's always this kind of tension that goes on between food flavor and food quality and what conditions you can apply to achieve your pasteurization. Beer used to be routinely pasteurized to destroy yeast and other things in the beer. It's much more common now to use another method we'll talk about in a minute, and that is filtration. So cold filtered beers, for example, are much more commonly used as treatment now as opposed to pasteurization. So throughout this section in the PowerPoint slides, there are typically some little summary slides that kind of throw everything together on one slide and give you a nice little summary of some of the heat treatment methods in this particular case, what some of their applications are, what some of their uses are, what kind of equipment you need to use to do these, etc. Another physical method you can use to control microbial growth is radiation. And the type of radiation we're going to be more interested in is electromagnetic radiation. Now, there's also another type of radiation called particulate radiation, which is alpha particles and beta particles. But electromagnetic radiation is basically massless energy that's created by fusion reactions. This shows you the electromagnetic spectrum. This represents the types of energy that are hurtling toward our planet that are being produced by the sun, and specifically the nuclear fusion reactions in the sun that fuse elements together of hydrogen, for example, creating helium. So the sun is basically burning itself out, and in the process of fusing those atoms together, part of the mass of those hydrogen atoms is being converted into energy. Remember Einstein said you could do that in equals mc squared. Energy and matter are interconvertible one another. In the electromagnetic spectrum, we have got shortest wavelength down here, longer wavelength here. And remember that wavelength is inversely proportional to energy. So these short wavelength forms are the higher energy. These are the lower energy. Greater amplitude of the wavelength, less amplitude of the wavelength here. So when we get down to this end of the electromagnetic spectrum, hopefully these forms of energy are not damaging or harmful to cells. If they are, we're in trouble, because we're constantly being bombarded by radio waves, infrared radiation. There's a lot of that long wavelength radiation we're producing as a society that hopefully will not have an adverse effect on cells. We get down to these forms of radiation, however, X-rays, gamma rays, various cosmic rays. Those are problematic. And then we get here in the middle, and that's kind of the sweet spot. These are forms of energy that are high enough quality to do some work, like photosynthesis, but hopefully not so energetic that they start damaging cells. Now, when you get down into the ultraviolet radiation down here, that gets a little dicey, because ultraviolet radiation is damaging the cells, but in a different way than these forms of radiation. These forms of radiation are called ionizing radiation, and these forms of radiation are called non-ionizing. And we'll talk about first ionizing radiation, and then talk about ultraviolet as non-ionizing radiation. Ionizing radiation passes pretty readily through most barriers. Now, things like lead shielding, for example, can stop ionizing radiation. But wood and glass and things like that, that radiation penetrates that barrier like it's almost not there. And it's absorbed by cells, and basically what ionizing radiation does is it breaks apart molecules. It can also produce a number of free radicals, and those free radicals are oxidizing agents, which can really play chaos with your proteins, with your cell membranes, with your DNA. And typically, the damage sustained by the cells kills the cells. These are lethal forms of radiation. Can we use ionizing radiation to treat things to kill microorganisms? Sure we can. Non-ionizing radiation is less intense. It's got a longer wavelength. And typically, it is blocked effectively by even fairly insignificant barriers. Glass, for example, can shield you from ultraviolet radiation. Water can effectively shield organisms from ultraviolet radiation. The effects of ultraviolet radiation and other non-ionizing forms is quite different from the ionizing radiation. We'll talk about that in a minute. Okay. One of the things that's really, really of critical concern in our society is the safety of our food. There are really two issues related to food illness that you need to be able to differentiate between. And those are food intoxication and those are food infections. And sometimes they are lumped together incorrectly and just referred to as food poisoning. So food poisoning is kind of a generic term. Technically speaking, you should differentiate and talk about a food intoxication, or food infection. What's happening in a food intoxication? The cells present in the food are producing a toxin. And we're going to talk later on about toxins and the various kinds of toxins. But one example of a toxin that can be produced by a foodborne bacteria is the botulism toxin. And that's deadly. But the key point here is that toxin is secreted by the cells, and it gets into the circulatory system, and it might then travel to very different parts of your body. And oftentimes the toxin has particular type of target tissues that it affects. So once you've ingested those cells, even if you kill the bacteria, the toxin still might have been produced already and be circulating in your body. What is the method of treatment then? You have to supply some kind of an antitoxin. Okay? Food infections are the result of living organisms, and in particularly the gram-negative bacteria and its LPS layer are the real culprit in most food infections. So if you effectively destroy the cells, you typically won't get the infection. If you don't cook something for long enough and some of the cells survive, then they can multiply and they can therefore cause an intestinal infection, for example. This is a slide that I'm not going to go through. I'm just going to have it there for your reference. But this talks about some major forms of bacterial food poison. Okay? And it talks about the microbe involved, the foods that typically are involved, and some very comments, oftentimes symptomology. And this doesn't really distinguish between intoxications and infections. It uses that generic term.
So when you're cooking food and you're trying to get microbes out of it so that you can feed yourself, like, are those microbes able to live within that organism when that organism's alive and still that organism not be affected by them, but just affect us compared to like a turkey?
Right, or sometimes what happens is the result of the food preparation. And so, for example, muscle tissue in meat is usually microbial free. But when they grind meat up to make hamburger, for example, if some of the intestinal bacteria get mixed in with that muscle meat, then your food can become contaminated. But it's indigenous bacteria that live somewhere in that organism, and then you're ingesting that. And if you don't heat properly enough, then those cells continue to survive, and then when they get in you, they multiply. Yep. There are a variety of methods that have been used to treat food for preservation. Historically, heat methods have been used, including canning, pasteurization, and cooking. Proper maintenance of temperature, refrigerating things, for example. Various preservatives and additives have been used to try to make food more safe, particularly salting, smoking, pickling. Those are all things primarily that are geared toward dehydrating the bacterial cell, and if not killing it, at least putting it in a suspended state of animation. Cleanliness is important, especially in the food preparation. But one thing that has had a difficulty gaining a foothold in food industries, it's making improvements now, is the use of radiation. And that's something I want to talk about a little bit, because there's a lot of misconceptions out there about the use of radiation in food treatment or irradiated food. You still don't see it a lot. But one of the things that's mandated by federal law is that food that has been irradiated has to bear this particular symbol indicating that it has been subjected to that particular form of treatment. The radiation that's used for treatment is ionizing radiation. One of the radioactive materials that can produce ionizing radiation is cobalt-60. And there are irradiators that incorporate cobalt-60 that you can put food in, and that exposes it to that ionizing radiation and very effectively kills the bacteria. Another nice thing about the irradiation is, at least for some foods, the quality of the food really is not affected at all. There are some foods that don't irradiate well, but other foods take it really well. It is much cheaper, it is much faster, and like I say, in many cases it really does enhance food quality. Why are people hesitant? Because it's been irradiated, okay? And they think that the radiation will persist. Now, granted, when things are irradiated, there are some free radicals that are produced, but those dissipate very, very quickly. And studies upon studies upon studies have been done that indicate there is no residual radiation present in irradiated food. And yet the public in general has this adverse reaction to radiation so much. We did such a good job of scaring people about radiation in the especially Cold War era, that there's just really this public kind of horror about eating this type of food. We'll talk about this a little more. So tomorrow, gather data.